Share. This one here. All right. Can everybody see my screen now? The cover slides?
That looks perfect. Thanks so much. And I really appreciate you being willing to talk to us today.
Yeah. Okay. Shall I start now?
Yes, by all means.
Okay, so good afternoon, everyone. Yes, so I'll be really happy to share with you in the next hour or 1.5 hour for our recent work on using the CompuCell3D platform to model this germinal center response, which is an important process in the immune system to produce high affinity, long-lasting antibodies for protection against future infections. Okay, here is the let me see, move this a little bit here. Here's the kind of the topic outline. I'm going to give give a little bit of introduction. What do we mean by germinal centers? What kind of cells involved, and what kind of phenomena we're really looking at. And then I'm going to talk about why CompuCell3D is really an excellent platform to do a modeling of germinal center response, and how we're going to do this with CompuCell3D. And importantly, I think we want to emphasize that in addition to using the CompuCell3D form, how we can actually export the simulation as separate files, so that you will be able to analyze these files, these data, simulated data afterwards for visualization, for all the statistics analysis. And hopefully we'll have some time to do some hands-on, or maybe depends on how your Python environment is set up. Maybe you can just watch me to go through some of these, the practice on visualization, what have you. Okay. So here is this graph shows what happens after we humans or animals are being infected by particular like a biopathogen, mostly like a virus. So when we first get encountered by some pathogen like antigens, what happens is that in the first 1 or 3 days, our body going to generate some what we call IgM type of antibodies, all right, and that lasts about like less than 2 weeks, and then after that, about a week, then our body generates some additional kinds of antibodies called IgG type, and these IgG type usually have much higher affinity compared with IgM to against these antigens we are being exposed to, and also when the infection get cleared, then the IgM usually go back down to basically baseline level. However, you're going to have a kind of long-lasting IgG actually always kind of in your body, ready for neutralizing, the same kind of pathogens when get second infection, and and when we do that secondary infection or exposures, then the IgM kind of response is basically about the same as previously. However, for the IgG response, it usually has a memory, so it can quickly launch a response which a much more amplified response levels. This is basically how what we call the adaptive humoral immune response works. So these kind of two type of common responses are mediated by B lymphocytes, okay, by B lymphocytes. Essentially, here is the kind of the basic biology about B lymphocytes, where the B lymphocytes are being generated, in the bone marrow, and then they after going through some intermediate process, maturation processes, the mature ones eventually will end up homing into what they call the secondary immune organs. This mostly including the lymph nodes that distribute through our body, and also the spleen, which you can consider as a big a big clump of lymph nodes, if you think that way. So these naive B cells, which means mature naive B cells, means they have never seen any foreign pathogens or antigens. So when they're being stimulated by antigens, they will terminally differentiate they will first proliferate and then terminal differentiate into what they call plasma cells. So the plasma cell is a kind of a terminal differentiated state of B cells, which is responsible for secreting all these immunoglobulins or antibodies, as we call it. So the B cell response in this case it can be really broadly divided into two different kinds. One is called T cell independent. That means the B cell itself will be able to launch a response in which once they get activated by these antigens, they will proliferate for a number of times, and then they will terminally differentiate into plasma cells. And these plasma cells are short-lived, so they are mostly secreting the IgM antibodies as I showed previously. So that's the T cell independent response is responsible for the red line you're seeing here. And then for the T cell dependent response, as I call it, it requires interactions with the T helper cells. And when this happens, the response is kind of different, because in these situations, it launches what they call germinal center response. And during this germinal center response, what happens is that the genes encoding for these antibodies will keep mutating, and as a result, only those mutations that's actually leading to high affinity antibodies will be conserved, while those cells that's actually having mutations that's leading to lower affinities mostly will get killed. So that's kind of a selection process, and also during this process, the antibody type kind of switched from the default IgM type to like mostly IgG type, right? And eventually these plasma cells that's coming out of this germinal center response is long-lived. They will go to actually home into the bone marrow and live there almost for the rest of the life of individuals. And also in this process, another output is the memory B cells, which is responsible for the recall response when the human body get exposed to the same antigen or pathogen the second time. Yeah, that's how basically so this T cell dependent response is mostly captured by this called germinal center GCR response. So the GCR response occurs mostly in the primary follicles, in the lymph nodes, or in the spleen, right? So lymph node, as we know, it's kind of like bean size structures, right? And then the germinal center is just a kind of it's a part in a slice of this lymph node, where under microscope it's about probably between 200 to 300 microns. That's basically the size we're talking about here. So under each microscope, each staining, basically the kind of the germinal center we're seeing here can be broadly, kind of divided into two zones, and what they call the dark zone versus the light zone. So dark zone essentially means you have kind of a more packed B cells, more dense, okay? And the light zone is less populated. That's what's going on there. And it turns out that the B lymphocytes will actually migrating between this dark zone, light zone for a number of times. And during these processes, they can expand, grow in size, and also going through all these mutations I talked previously, eventually going to churning out plasma cells that secreting high affinity antibodies. So that's what the germinal center response is all about. So here it's kind of a little more details what's going on. So the B cell will be actually migrating back and forth between dark zone and the light zone. So in this figure, the dark zone is on the left and the light zone is on the right. All right. So this migration is driven by chemotaxis force fields. In these situations, there are two chemotaxis factors here, CXCL12 and CXCL13, which forming these opposing gradients as you see over here. Well, the CXCL12 is being actually secreted by they call the CRC cells, like these star cells in the dark zone, and then these CXCL13 cell, molecules is being secreted by the follicular dendritic cells, as you see over here in the light zone. So depending on whether the B cell expressing the receptors, for these two chemotaxis factors, chemical factors, then that will determine the B cell moving, to the light zone or to the dark zone, right? So what happens is that once the germinal center is launched, the seeding at the activated seeding B lymphocytes will first actually interacting with the follicular dendritic cells, FDC cells in the light zone, where in this interactions, as you see over here, the B cell, which express the B cell receptors, will be able to recognize the antigens of the epitope that's being expressed on the surface of these FDC cells. So once these BCR recognize these antigens, all right, they're going to engulf or internalize these antigens into the inside of the B cells. And then these B cells will re-express, okay, these antigens on the surface of the cells with the help of these MHC2 molecules. So that's in this situation, the B cell become the antigen presenting cells, and also how many of these MHC2 peptides, being expressed on the B cell surface, really depending on the affinity of the BCR for these antigens presented on the FDC. The higher affinity, the more internalization happening, and the more the MHC2 antigen complex going to be expressed on the B cells. So once these B cells, after interaction with the FDC, will be able to then, interact with some the T helper cells, okay, the TFH here, and also in the light zone. And when this happens, it will trigger a kind of a number of responses, including the TCR response, and then the T cell will be activated by the TCR. In turn, will express CD40 ligands as number of diffusive cytokines to activate the B cells. So this is really mutual activation process. As a result, if the activation by T helper cell is strong enough, then these B cells, which tend to have a higher affinity for their BCR against these antigens, will be selected, positively selected. Okay, once they're positively selected, that means they will not die. They will not go through apoptosis, which is their default fate, and they will be surviving. And also, they will be starting to express CXCR4 receptors, which is for the CXCL12 chemokine. As a result, these B cells will start migrating into the dark zone. And in the dark zone, well, actually, when they get activated by T cells, they also trigger the cell cycle. So as soon as they reach the dark zone, they will start to proliferating for like a number of times. I think the number is between probably 1 division to up to 6 divisions. On average, probably will divide by 2 or 3 divisions. But the number of divisions really depend on the degree of activation by the T cells, which in turn depends on the BCR affinity by the for the antigens. So during the division process, every time it's kind of divides, it have a chance for mutation. So for the daughter cells, so the mutation always can be good or bad, right? But for some really bad mutations, such that it will not be able to form BCR, those cells will die. So apoptosis also happens in the dark zone. But most of these, what they call somatic hypermutation, will be determined whether they're good or not by having these cells moving back to the light zone. Okay, so after a number of divisions, then these cells will lose the expression of the CXCR4, and as a result, they will be being attracted by CXCL13 back into the light zone. And in the light zone, they're going to go through that interaction with FDC and the T helper cell again, as I just mentioned. So that this whole cycle is kind of probably going on for like 2 weeks or probably even longer than that before the the germinal center really kind of disappears when all the pathogens are being kind of eliminated. So really, this is kind of when I first learned about this process, I thought, okay, this is really a kind of a nice project that using the CompuCell3Ds to allow you to be able to model this process both morphologically with the cellular behaviors, as well as using kind of intracellular gene networks to drive these cellular behaviors. So here, what we're going to talk about is actually the project, which the paper we just recently got published in the Journal of Frontiers in Immunology. So the work mostly, the one we publish actually is kind of a much more complex than the model we're using here for demonstration purpose, just for your information. Okay, well, but I'll through the process, the remaining talk, I've talked about some differences between what we'll be using here and what we're actually modeling. Okay, so here. The first thing that you want to really model the CCR for me is that in addition to the cellular behavior you're seeing there is how do you model this kind of intracellular signal transduction and the gene regulatory network, which is being stimulated, driven by cell surface receptors, in this case, including the BCRs and CD40, and also like CXCR4, 5 receptors, what have you, right? So our understanding about this network is still incomplete. So the first thing is to simplify. We only basically retain the molecules, the genes, which we think are really crucially important for guiding the behavior of the B cells. And so with the simplified network, we'll be able to implement that in Tellurium in the CompuCell3D process. And then the behavior or the actions of this gene network will be linked to the cell behaviors to guide their migration or cell death, what have you, as you'll be seeing. So here are some of the key biological events that we're simulating here. First of all, the major cell type will be the B cell, of course, and the FDC, and also the T follicular helper cells in the light zone, and then the CRC, which is expressing the cells CXCL12 in the dark zone, and the B cell chemotaxis will be basically dictated by the CXCL12 and 13 gradient, and then the kind of the expression of the respective receptors for these chemokines. So in terms of the different phases in the lysome, we'll be modeling the interactions of the cell contact between B cells with FDCs, okay, and the consequence of that contact is that the B cell will be able to take the antigens on the FDC and re-expressing them with the MHC2 at the tag, and then the B cell will next interact with the helper T cells, where it will trigger the CD40 signaling, and depending on the PMHC2 expression density, then that determines the probability that these B cells will be positively selected or not. Okay, and then that in turn determines their survival, dark zone reentry, as well as initiation of cell cycles. Of course, for those ones that are not being selected, will die in the light zone. In the dark zone, what's happening will be the B cell will start to divide, go through mitosis for a few times, and during that process, we'll be randomly mutating the genes encoding for this immunoglobulin, and then there's a chance for this cell to die as well if the mutation deemed to be just too severe, such that will not form a kind of BCR expression on the cell surface. Yeah, so that's basically the key events we're modeling. Let's reflect the B cell biology here. Okay, so here is just to kind of give you a preview what's going on, our simulation result for this process. All right. So again, on the left is the dark zone, on the right is the light zone, and then we're starting with like about 200 B cells in the middle, and to see how they actually, kind of the migrating between the two zones, and then as their color changes, so the color of these the B cell reflect the affinity. All right, so the blue is kind of like low, and the red is high. And as you will see, as the time goes by, the germinal center evolves. Essentially, the cells remaining will be expressing high affinity, means color will be moving towards yellow and the red. So. I probably need to advance this because it probably it's like a minute or two something. So as you see, these cells are moving, and further to the lysosome, most of them get killed because their affinity level is so low. So only some of those actually by chance survive and move to the left, and then move to the dark zone. They're starting to proliferate and turning out more B cells. And these B cells will again, once they terminate their cell cycle, they're moving to the left, to the right, which is the lysosome again. And in there, they're being selected. Some get killed, most of them get killed actually, and then only selected few get chance to move back to the left, which is the dark zone. Okay, and this process just keep going, and in the meantime, the number of the B cell will actually growing as a result. In addition to improving affinity, the antibody affinity. So let me. I try to drag this stuff, but it doesn't work. It only work when I stop this whole thing. Let me see if I can do that way for now. All right, so let me move this. You still see my PowerPoint here, right? Yeah. So what I'm going to be doing is I'm going to dragging this actually, as you see. Yeah, you see this, the colors start to going towards the green, yellow, and red. That's indicating these cells their affinities are actually starting to improve. And the number of cells become more and more as they proliferate. And eventually they'll form a structure that's very similar to people see under the microscope, where you have a kind of a dense population on the left, which represent the dark zone, and they have a kind of less dense area on the right, representing the lysosome. Okay, so now let's go on presentation. All right, so here it's kind of a few screenshots capturing what's going on there as the germinal center evolves. Okay, started with these are what we call seeding B cell clones. Usually for germinal center, they can be seeded by like 50 to 200 B cell clones. All right, but during the process, most of them probably will get killed, but only those ones happen to have like a higher affinity slightly, or the others will survive. And during this process, their affinity will keep improving until you reach the end of this. And eventually, as the infection clear, these cells will also actually eventually disappear. The germinal center will just regress. Okay, we're not showing that here. We're just showing this rich steady state. Okay, so for our purpose here, we're using this GCR demo .cc3D for illustration purpose. Essentially, we, as you probably already familiar with the structure of germinal cell, the CC3D, right, project, where you basically have, like 4 different kinds of files, which is the main one, the XML files, and then the Python, which basically has all the details of the script, and then you can have a PIF, which actually defines the locations of some cells to begin with. And then here we also have you're provided, right, these some additional files, which is kind of the Python script that you'll be able to run outside of the CompuCell3D to analyze the simulation data that's being saved into this called cell data folder. Okay. So I'm going to go through some of these key things here. So in the XML file, we define this germinal center space. So for the demo file you're seeing here, we're only using for, because we want it to be fast. So we're only using a 2D space, which is about 100 by 100 pixels. Well, in the actual paper we published, we actually make it into 3D, which is about the size of 250 by 200 and by 11. So as I said early on, that's probably the average diameter of a germinal center is about 250 microns. So that's pixel. This kind of dimension, try to recapitulate that. But again, you probably, for the Z stack, you probably will go up to 50 or even higher than that. Again, that will be too slow for our simulation. So again, in our published work, we just using 11 layers for the Z dimension, just to make the simulation just reasonably fast. Yeah. And then in the demo file, we actually in the PIF PIP files, we define a wall surrounding these the space where we're simulating, and so it turned out to be not necessary, as in our published version we get rid of all the wall anything. So basically all the B cell will stay in place because they're always being chemo-attracted by the CXCR CRC file cells in the light dark zone, or the FDC in the light zone. So they never actually leave out of the picture. So actually, so we don't really need that. Eventually for our final version here. And then we're seeding the cells here. In the demo version you're seeing here, we're basically seeding the CRC in the dark zone randomly, and also we're seeing the FDC and T cells in the light zone randomly. But these cells, once they're being seeded, they're fixed. They don't move, okay? They don't move. That's pretty much true for CRC, for FDC, and actually for T cells, they actually in biology, they still move. But here, for simplification, we're just immobilize the T cell as well. So, but in the actual paper we published, we actually position these cells in more well-defined locations. I will see in the next slides. For example, in the demo here, these CRCs, the smaller the T cells and also the FDC, we just randomly kind of distribute them in the dark zone here or the right zone here. But while for the published work, we want to make the more controlled settings so that we can reproduce some of the results. So we kind of place them more like the way that we expect that pattern. That's what's happening in the germinal center. Yeah. So then in terms of in this demo files, we're just start to assign some antigens to FDC. FDC are antigen presenting cells, right? They actually function as, like, say, macrophage. So they engulf these antigens and then kind of break them up into epitope and express them on their cell surface. So for example, we're just using the ATGC letters, alphabet, to simulate the antigen sequence. All right. So we kind of pre-assign. Okay, let's say this FDC is only hosting an antigen that's had a sequence of AGTCT, for example, right? And that's the antigen sequence that's the antibody gonna need to improve to be against for. And then for the initial B cells, which is the B cell clones that's seeding the germinal center. Okay, in these situations, or just you can do whatever you want. Okay, so in this situation, you can randomly assign these 5 letters so that they either match or they don't match with the target sequence, antigen sequence there. Okay, so this what's kind of this line of code is doing. Essentially, it's just picking, let's say we're seeding the germinal center with 6 B cells. Okay, for each B cells, we're just randomly picking from the 4 letters here, and then forming this 5-letter sequence, which is kind of the starting antibody sequence, yes, for that particular B cells or B cell clones. And then I think here what we're doing here is that also we're seeding the B cells in terms of locations. In this case, just randomly, we're putting this in the middle of of the germinal center to begin with. That's what this chunk of code is doing. And then this one is actually assigning those random sequence to each CDR B cells, there. Okay. Yeah, and then here this chunk of code is about that. Once a B cell, a new B cells is being right. So actually, I want to point out this line, right, is the line actually producing this the initial CDR B cells for this situation. Okay. And then once the B cell is being created, and then you want to assign them things, right, like the target volume, the lambda volume, and then also the B cell, the one that's responding to the chemokines. So you're gonna assign them to the add the chemotaxis CXL12 to the B cell, and then set a lambda. To begin with, like say you were saying all moving to the left or to the right, okay, and then also for the cell CXL13 as well, and then at the end of that you want to add that the B cell network model, attach that to that particular B cells. So yeah, so the chemotaxis field is defined in that XML files, as you see here. Basically, we're saying here we have a field called the CXL13, and that one is being secreted by the FDC cells at a rate of 0.1, and that one has a diffusion constant 1 and have a decay constant 0.01, for example. And similarly, the CXL12 is being secreted by CRC in the dark zone, and that has own parameters for secretion as well for the diffusion and decay here. Okay. And then, okay, so here is the kind of the field that's in our published paper, where you can see that for the CRC, which secrete the CXL12, have this nice gradient, okay, at higher concentration on the left, dark zone, and this lower concentration on the right in the light zone. And the CXL13 had the opposite gradients, as you'll see over here. And then here is kind of the simplified gene network model, which have expression, which responding to signals from for the T helper cells through CD40, and also responding to the FDCs through the BCR, and then the one responding to FDC will be able to re-express that antigens with the MHC2, and they form a complex called a PFHC2. And that complex will be able to activate the follicular, the T helper cells, which interactivate the CD40s. And then the downstream signals is complex. It's involve a number of intermediate signal molecules, and then also kind of transcription factors that driving different functions of the cell, including like AP4, MYC, mTOR for cell growth, and then the BLIMP1 for plasma cell differentiation, and then for FOX1 responsible for expressing CXCR4. And then other fates like the somatic hypermutation, and the apoptosis we have. Okay, so just here, just a snapshot of part of the Tellurium gene network model here. So in terms of the B cell interaction with FDC and T cell in the light zone, so we're using the neighbor tracker plugin, and will determine whether or not the B cell is in contact with an FDC or T cells. So in the XML file, you have this plugin declared, and then in the Python script there, here it's kind of basically a few lines, evaluating whether the B cell is in contact with FDC or not. Basically, you kind of first using this get function to get a list of the neighbor list, right? And then you kind of report back what type are they, okay, and cell type, right? And then you're going to ask whether number 2, which is our FDC cell type ID, is in that list. And if that's true, then we assign this variable touch FDC as true. And then we're going to be a bit, then the next thing is that if that's true, number of things going to be happening. Yes, that means at that moment, the B cell is in contacting with FDC, all right? And so, and then what happens is that the first thing that it's going to read the target sequence that's being held, in the FDC in there, and then that sequence going to be being read by the B cells and then compared with the BCR affinity or the BCR sequence there, and then to determine how the affinity of the B cells, because their BCR, so BCR essentially are surface expressed immunoglobulin, right? So as the B cells, their immunoglobulin being produced, but they're not being secreted. They're only expressed on the cell surface. Once the B cell become plasma cells, then those immunoglobulin will actually will be secreted. Yeah, so that's the kind of the difference here. They're all being encoded by the same genes, but the fate is different in terms of location. So in these situations, they're comparing basically the target sequence, in the FDC with the actual BCR sequence to determine the affinity. Okay, to determine the affinity. And then the next thing is it will determine whether it's interacting with the T cells. In this situation here, what happens is that for B cell to responding with to T cell, it first has to contact with FDC to express that MHC2 antigen complex. Okay, so you have to first say, okay, if only one that's true, I'm going to doing the following signalings. So that's what happens in these situations. Then we're going to use the affinity score determined over here, okay, to determine the chance this B cell going to be positively selected by the T helper cell or not. So using that affinity score divided by the maximum score give you a percentage, right, chance. And that percentage chance is going to be compared with a random number drawn between 0 to 1. And then that's going to determine, all right, they're going to survive or not. If they're going to survive, they're going to turn on the NF-kappa B signaling, okay, which is the K7, which is determined by the K7 defined parameter in the Tellurium model. Okay, so and then also it will turn on the expression of CXCR4. So that will be assigning K1 instead of being 0 to 1. So the CRCR4 will be highly expressed. Okay, and also in this situation, we can turning off this death timer that is ticking before they encounter the T cells. Now the death timer is reset to 0. So that means this particular B cell at this moment is not dying. All right, they're surviving. And with the CXCR4 re-expressed, they're going to be moving back to the dark zone. And with the kappa B being activated, they're going to activate the cell cycle. Yeah. So here, yeah. So once the cell moves to the dark zone, all right. If they're in addition to being kind of dividing, if their kappa B level drop to a particular threshold levels, we're gonna say, okay, the cell will no longer actually be surviving. In that situations, okay, the cell need to die. So in that situations, we're gonna set the cell death timer to 0, and then we're gonna delete FCBM model from the cell, and then delete the cell from the simulation. So that's how the cell death timer is being implemented. In terms of the cell cycle, so the NF-kappa B activates MYC, which is transcription factor, and then MYC activates AP4, which is another transcription factor. So we determine in this case, once AP4 reaches a threshold, it's going to start the cell cycle. So in this situation, the cell is going to grow exponentially with a particular double time. So to give you a number, usually these cells they double pretty quickly. So basically, their cell cycle only about 5 to 6 hours. That's basically very fast in terms of mammalian cells, especially for humans. Okay? So they double every 5 to 6 hours. And so the volume will be growing based on that exponentially, as you see over here. So our target volume will keep updating based on this rate over here. And then here we're saying if the cell volume reach a particular threshold, in this case doubles, it's about 32 because the baseline is 16. And then if the cell volume doubles to 32, and then it's gonna initiate that kind of cell division. In this case, right, we're gonna have a new cells that's being made over here. And then the new daughter cell will copy everything from the parent. Okay, so now you have two cells instead of one. Okay, instead of one. So, and that inherits actually in the published version of our model, actually we gave some stochasticity. So there's kind of a randomness like distribution in terms of the molecules, how these molecules are being allocated to the two daughter cells based on this binomial distribution. Okay, but for this demo, we don't have that. We just a straight copy of the parent cells. And also in this process, we're in this demo file, we're kind of doing this somatic hypermutation. So basically, remember, each cell has like their sequence of like file letters, right? We first determine among the file in which position the mutation is going to be happening, and then the next one is among the 4 letters, which one is going to be choosing to mutate to. So essentially, this chunk of code is about that. So here is determining the index among the file letters in a sequence, which one to choose from, and then here this one is picking randomly, among the 4, actually among the 3, we're not mutating to the same letters. Okay, so we are picking among 3. Yes, from the alphabet to mutate the sequence into. And okay, so here is something else where because we want to do so much analysis afterwards after a germinal center simulation, so we end up actually almost saving each individual cells as a particular frequency. So in these situations, we're actually saving all the variable levels for every single B cells at like a 15 the Monte Carlo set MCS in this situation. So we save them into a simple TXT files, simple text files, and by naming them as follows. So the file name will follow this convention where A will be represent the generation. So the seeding B cells will have generation 0, and then followed by a high underline dash, and then by the mother ID, and then followed by the cell ID of the B cell itself, right? So as you can imagine, for the seeding B cells, their generation 0, their ID, they're both their mother ID and the cell ID are the same, right? Because they don't have a mother in this situation. But then for these following generations, the generation number going to change, and then this one tells it's coming from this cells, and then this is the ID for that particular cells. So on and so forth. And within each text file, once you open up, it has a header. So the headers tells you all the variables that's being saved, including MCS, the cell ID, mother ID, MHC2 antigen complex level, lambda 12, 13, and all the variables basically in the gene network we're modeling essentially. All right, so every cell keep recording until that either that cell dies or that cell divides or the simulation terminates. Basically, that's how we're doing this here. Okay, so the next thing is that's about like using that the Python file outside of the CompuCell3D to analyze the simulation data. Essentially, that Python file will be reading all these TXT files, and then they're gonna recursively to find the lineage, essentially to remap up that's kind of a binary tree, as we'll show you later. And then they kind of combine the cells in a single lineage branch, and then you will be able to select the variables you want to see to display the behaviors for that particular lineage branch. And eventually going to be because once these cells, these text files, they're being saved as string, right? So you have to convert them into like numericals to be able to analyze or display them, right? Okay, so here just give you some idea of the we call the read file function, which essentially reading each TXT file line by line, and then save them into a variable we call lines over here. Okay, so this one will be called for each cell or each file saved there. And then this is the function we call retrieve lineage branch function here. Essentially, I'm not going through the details. Essentially, it's about like giving a cell ID. How do you trace out every subsequent sibling cells? Okay, as offspring of that particular cells. Yeah. So this about again kind of about actually using like say the cedar cells as the at the top, and then recursively getting their lineage cells. For example, in this situation, if you feed the cell ID 62 into that function, it will be able to trace out all these binaries nodes over here. Each node represents, of course, a B cells, right? And so at the terminal ends, these are the ones that either die or it represents the end of the simulation. Okay. Okay, so here it just give you some. We're going to be seeing this on the fly. So, like the example output, like say when I run that kind of lineage retrieval function, how many lineage will spit out? Okay, basically it counts every pass from starting from a cedar B cells all the way to the leaves, basically all the way to the leaves. All right, so that's represent every single branch. And then you can pick a branch which means contains all the cell IDs along that branch, and then be able to combine all the cell data in this branch, and as a big chunk of data set, and then be able to kind of tell them which variables you want to display for that particular one. Like say I want to I can type like xcon, ycon, which is xy coordinates, and also like MCS or other gene variables you want to visualize. So this is just the kind of data type conversion stuff where from the string to kind of float numericals, and then eventually you can use these scatter plots for simple visualization of the trajectories of these cells. So for example, using this, if I'm just using the XY com and also MCS, I'll be able to plot the trajectories starting from one cedar cells up to number of kind of back and forth migration between the two zones, and after a number of time of divisions, and just following with that one single branch, and what happens to that lineage branch. So in this one, it starts from here at the cedar cells, it's back and forth a number of times, and then ends here. For this particular branch, the cell fate is apoptosis or mutation death in the dark zone. Okay, so the color represents the MCS time. And then, for example, this one, it ends in the light zone, but it ends as a plasma cells exiting the simulation. And the middle one, that branch end up as start from here and then end up as cell death in the light zone. And with this, you can also do this kind of time course where the gets cut off. So the x-axis is the MCS multiplied by 100, right? So it's about the time. And in this situation, we're also kind of tracing one particular B cell lineage branch. And as you can see that here, the panel A represents the X coordinate. Basically means the high numbers meaning the cell reaching the light zone, the low numbers meaning cells is getting back to the dark zone. Okay, so it's doing this and forth, back and forth. And then you can see that when the cell getting to the dark zone, their volume kind of up and down doubles and back down double and half again for a few times, represent the number of divisions or clonal expansion, or sometimes called a proliferative burst that's going through at the time they're spending in the dark zone. And then they're moving to light zone and then moving back to dark zone. And so every time this number kind of random, but it's statistically supposed to be proportional to the antigen affinity. To the antibody affinity they're being expressing. And then as time goes by, the cell generation will be rising. And also you see that for this particular case, the affinity keep kind of improving in this case, because this is the one I think this is the one that is this one on the right here. That's the one end up with a plasma cells. So that's why it's kind of the affinity keep rising until it reach a threshold. Then we decide, oh, that cell will become a plasma cells. So, and then other variables, genes that's being cyclically expressed. For example, the CXCR4 is being cyclically expressed. So we determine the cell moving back and forth between the light zone and dark zone here. And with those TXT files, another thing you can do, you can recapitulate these images, because with the CompuCell3D, you cannot save every single variables at a particular time step. That's or intervals. That's going to be cause a lot of space and also potentially slow down the simulation. So here with this TXT files, you can recapitulate, like a snapshot, like doing like a immunohistochemistry, like staining for particular marker genes and how they're being expressed at different zones. Yes, as you see here. And then with that, you can do these analysis like you're doing single cell analysis, like for dark zone versus light zone cells, how they're different gene expression, different for these volume plots. Yeah, you can do all these kind of analysis. Right, and then here again, this is showing some results. We're presenting our published paper. We're seeing that the germinal center B cell population as a whole, how that's the affinity overall kind of evolving. In these situations, it's the average affinity keep rising until it's plateaus. Okay, and then here these other color lines represent the 75, the interquartile or the 95% interval, or the maximum and minimum affinities that's for that particular population of B cells. And those dotted ones are the ones actually eventually end up as the plasma cells. And then the panel D shows really that the number of B cells, how that's kind of increasing over the time and eventually reach kind of steady state. And how the cell in the dark zone versus the light zone are actually kind of and also their ratios. And according to literature, basically the B cell number in the dark zone is about like a 2 times, twice as much as in the light zone. As you can see, that's at the beginning when the cell number is very small, this ratio kind of fluctuates, but eventually it's kind of reach a steady state about 2-to-1 ratio over here, as you read on the right y-axis. And another thing interesting here is that the Germinal Center can start with like 100 B cell clones. So each one has slightly different antibody sequence. All right, but at the end of the Germinal Center, usually only a few or single clone will be dominating. And our simulation exactly kind of recapitulate that. For example, in panel F is more clear. You started with like 200 clones. Okay, each one has 1 cell, so you have 200 cells to begin with. But as the Germinal Center going on, most of clones will be eliminated, and only one in this case eventually dominant. And then there are a few less dominant ones. Yeah, but essentially that's kind of consistent with the literature that Germinal Center kind of evolved into some dominant clonal dominance phenomenon. All right, so to summarize, really, the CompuCell3D model we're using for the Germinal Center response using the CompuCell3D platform. We believe that's kind of quantitatively recapitulate a lot of the key phenomena, both morphological as well as functional events for the GCR, and then a lot of the post-simulation analysis can be really performed if you save each individual cell data and then be able to kind of analyze them afterwards. Yes, and of course, a lot of things that's need to be improved in terms of this Germinal Center response. But for the lack of time, I wouldn't go over those here. So basically, I really like to thank my co-authors for this paper and project. Essentially, Derek Mu, he actually a high school student. He just graduated this year, and he's going to Carnegie Mellon for a computer science, degree there. And he's the really one that's actually behind the scene and doing all these the heavy lifting Python coding in the CompuCell3D, as well as in the main Python analysis code. So that project has been kind of this project has been going on like over two or 3 years, and actually Derek, he's the one actually presenting this in the last 2 CompuCell3D workshop. So this year he's not available, so I'm the one really kind of stepping in to the presentation. And then Chris is kind of my collaborator at Emory. He's really a B cell immunologist. And then Dr. Minsky, which the director for our super fund funded by NIEHS. In that study, in that project, we study like how environment pollutant like dioxin impact these B cell response. So now with this kind of physiological model, the next step we're doing is trying to implement how these environmental pollutant dioxin actually impact this GCR. One thing that has been found that's the dioxin maybe actually inhibits the proliferation step of the Germinal Center response. And one more thing is that epidemiological data has shown that really the dioxin level in our circulation is kind of inversely correlated with the antibody titers in our circulation as well. Okay, so with that, I think I'm kind of done with the presentation part. So for the remaining time, I think we can do some more interactive with the model codes that's we provided in the previous years, like in the because our paper wasn't published yet, so we're not really, we're holding back on the you know, the CC3D script. So this year, we're able to actually share with you a kind of intermediate version of this model, because the final version of the model will be in the GitHub. All right. So, but still, we cannot really use here. It's just too slow to simulate. So that's why we're using this kind of an intermediate demo version, just to give the idea how we model this process and how we can analyze the simulation data. So, James, are you there? James?
Yeah, I'm here.
So, I mean, what should I be doing? I mean, are there questions posted there?
Yes,
or
people have questions they can ask you.
Right. Maybe at this moment we can, you know, answer some questions before we get to the the exercise.
Sure, please go ahead. If there are any questions from the audience, please unmute and ask. And if not, we can do the demo. I was having some trouble getting it to run because of the path names, but Sharon has it running on her Mac, so Mac people should be able to run it without a problem.
Okay.
So go ahead. Go ahead with questions, please.
Yeah. Any questions about the biology, about implementation?
You went through it quite quickly, but this has been a deep interest of mine of how to integrate Tellurium data into these CC3D models and what data can be gleaned.
Yeah.
I don't even know what my question is because you've presented it, but it's the first pass going through. So I see that you loaded the Tellurium data. The thing that was most impressive is that you could predict expression of these various targets in the pathway spatially.
Yeah.
You mean,
okay.
Yeah. How did
you mean like this one here, right?
Yeah. Yeah.
Okay. Yeah.
What are some of the if you could just summarize the how CC3D can work with Tellurium data for
you mean actually how
behavioral insights?
You mean the simulation, or you mean the afterwards post simulation analysis?
Post simulation analysis, yeah, because the signaling pathways don't have any dynamics to them in their schematics, and I believe it's CC3D is enabling dynamics to be shown. But I guess I don't really know how the parameters are being established. I guess Tellurium is establishing the parameters.
Right. Okay, yeah. So I think maybe let me just I think it's better. Can you hold on this when we show this CompuCell3D script? I probably can better explain that to you.
Okay.
Yeah. Other questions from the audience? Okay.
I had an earlier question, which was from your earliest images. I didn't see a description for cell death, but you modeled it a bit. I was only thinking of clonal expansion, but I wasn't seeing where cell death came into play. But that was certainly a component of the model.
Yeah, okay. We do have that here.
Death timer.
Yes. I think that's kind of there's a two kind of cell death here. I mean, they're all apoptosis, probably mediated by different apoptosis pathways, but cell death happening in both the dark zone and also the light zone. And according to I think a few more recent papers, they're probably happening at a very similar rate. As originally people think mostly B cell are just dying in the dark zone because of a negative selection, what have you. But it turned out that because of the hypermutation there, okay, can be so disruptive, such that some of the immunoglobulin molecules, when they're being expressed into the protein form, they cannot really fold well to become a BCR being expressed on the cell surface. It turns out that they have a mechanism to eliminate those cells. So those cells will go through apoptosis there. Okay, so the way it's not we're not modeling in this demo file, but in our published paper, what we did is that based on the literature, I think we give a 1/3 of a chance for these really damaging mutation. All right, so we encode this like 1/3 of a chance every time a B cell divided into two daughter cells. One of the daughter cells will, like both daughter cells will have a 1/3 of chance that's just disappear because of being killed because of damaging mutation. Yeah, so that's the way how the cell death is being implemented. So basically, of course, in the simulation, just delete that cells, right? Yeah, delete. That's it. And then for the lysosome death. Based on my understanding about this germinal center, is that once the B cell kind of migrating from the dark zone to the light zone, once the decision to migrate back to the light zone is made, then their death timer will be already started. All right. So that death timer is kind of a random thing. It has probably an average, like a death time, what have you. But then. So the default fate of those B cells returning to the light zone will be death, unless they get interacted with FDC, then the T helper cells, and get rescued from that. Okay, then those cells will survive. So otherwise, those cells will just die. If they don't get access to T cells, that's why one of the reasons they're saying the T helper cell is the limiting factor for the germinal center response for generating high affinity antibodies there. Yeah, I don't know if I answered your question.
Yeah, you did. I guess since I'm still on mute, I'll just ask one final, which is, what impact do you believe that this publication has had in the field? How do you think this dynamical modeling has moved things forward?
Oh, you mean what impact? I think with this model and probably some future iterations, because during this review processes, we learn something new, and we know some of the limitations we know, and but for simplicity we didn't put it in there. But with this model and also future iterations, we believe we can explore a number of things in terms of kind of vaccination efficiency, all those stuff. Like say you can have different kind of antigen presented in the germinal center, right? Then your situations, the B cell can be confused, right? Which one is optimizing for? And then you can do things like they call it feedback stuff, where when you do have plasma cell formed that leave the germinal center producing antibodies, and those antibodies will circulating back into the germinal center, starting blocking the antigens, and then preventing the remaining germinal center from responding further. Yeah, so those are all things happening. Yes, I think with this model can help you explore a number of things, I believe, at least theoretically. Mm-hmm.
Thank you.
Okay. Now, okay. So let's actually to look at the simulation here, right? So essentially, the file you are given is two zip files here, right? The GCR demo, which the one that's being run with CompuCell3D. So once you unzip this, inside you have the routine structures you're seeing with a CompuCell3D project. So we're going to be opening this one here. I guess but at this point you all know how to open this, right? You just go to project, and then you say open project, and navigate right in your Explorer here to that particular files. So in my situation, I'm in the Google Drive here. I got a lot of stuff here. Yeah, you just go migrate to your directory, where you have this GCR demo folder, and then you just open GCR demo CC3D, right? Yeah. Okay. So once you have that, you have all these structures now, right? So just open the main Python file here on the bottom here, right? So one thing probably you need to do is, because this one is saving all these variables at particular interval, right? For each B cells as a TXT file. So you have to tell them where to save. In my situation here, on a Windows machine, there's 2 places. I save that into a folder I call cell data on my desktop. All right. And on Windows machine, if you proceed with this sign, if you find that desktop, and then but you have to manually create a cell data folder on your desktop. Okay. So basically, if let's say I'm kind of what do I have here? What do I have here? Yeah. Do you see my desktop here? You do, right? Oh, you don't.
Yes.
Yeah, you see the cell data folder here, right? So basically, just right click on I don't know about Mac or other. You just say new, generate a folder and name it as cell data, and you can name whatever, but you have to change the name in your script as well. So D is a capitalized D. Right? And so once you have that created, then this is one place that's saving the headers, and then there's another place that's actually saving the data, the simulated data every 15 MCS in my case. Okay. So once you have that created, you should be able to run this. So I'll right click, say open in player. Let me see if it runs. Oh, that's interesting.
So I had to add a line saying sample frequency equals 100 at line 13 in the code.
Okay. I think, did I change something? I think I deleted it, but then I turned out not using it. Oh, I know. I know what's going on. Sorry. That was running. Okay, sample frequency.
But if you set that equal to something like 10 or 100 in the...
I had it previously, then I found out it's redundant to the save frequency. So I decided not to use it. And it turned out that there's one, okay, SAM sample. Okay, I think here is the place. Somehow I changed this. I think this is the only place that's being used. You can change this to hold on, let me see. That's weird. Okay, let's do this instead. I think previously I have this defined as the same one as this one. Just create another a variable called the sample freq equal to 15. I hope that's gonna save the situation. Okay, so now it runs. All right, let me just say I'm gonna change this to CSR4 and create another one. Gonna call it the affinity score. No, this can run. All right. So to repeat what I just said, somehow the file I send you, I delete this one, just adds another line called sample freq equals to 15. Yeah, that will work. Sorry about that. I think I made some change in the last minutes, but I forgot to change the other one there. And then that's caused issue. Do we have questions here? Yeah. So it's supposed to be running like this now. Let's see how many people actually. There's a way we can survey our students to see if they're okay. Oh, that's the one. Okay. Oh yeah, it's okay. That's nice. All right. Just
I got it to work, but I had to edit that the directory.
Okay, the folder. Yeah. So you need to do this, add this to the script, and also create a folder on your desktop if you're using Windows, and then that should work.
Sharon says that works on a Mac too.
Oh, the same. Okay.
Creating this, you have to create the directory, cell
Right,
data.
you have to create a directory, yeah, called cell data, D capitalized. Okay. Yeah. Okay. Let's give some time. Folks still. Right. So for those folks who actually got this running, you can start observing how these the color ones, if you pick the, like say in my case, CSCR4 and affinity score, okay, and these cells will be just moving as you see between these two zones, and the CSCR4 need to be turned on or become red or yellow for them to be in the moving to the dark zone. Okay, for those ones that's actually losing those colors are the ones actually, as you see here, is moving to the light zone there. Okay, and then for the right panel here, as you see the affinity score, those ones will be changing as well, because every time a device, there's a chance of mutation, so that affinity score going to be changing as well.
Can you explain that one more time? So the colors in the CXCR4 level are based on the chemotaxis of CXCR4. But what I guess I don't quite understand is that the cell type IDs are. I thought the small squares on the right were T cells, and the larger ones were dendritic cells.
Let me explain. So these immobilized ones here, the CRC cell, we don't need to care about, right? So the one on the right, the bigger square are the FDC cells, and then the little ones are the helper T cells.
Right.
Okay.
And how come we aren't seeing those colors? based
So
on what
those they don't have color in this situation, right? They're just irrelevant in terms of I mean, when you view them as view the CRCR,
Oh, right. Okay. Yeah, that's
if you
what
go
it,
back to cell field,
yeah.
they do have their own color. That's a different thing, right? Yeah. So just to probably to go over, okay, so here is the Tellurium gene network model in this demo version here, okay? So as you can see here, the CXCR4 is being synthesized with the zero order reconstant K1, and then it's being degraded with the first order reconstant K2, right? So in this simple version, we're just by after the cell will be contacting with the T cells, we're going to be determining whether the K1 going to be 0 or some positive values. So that determines whether CXCR4 is being expressed or not. Okay. So as you can see, if you search for K1, for example, in this one here, right? Come on. Yeah. See here? In one place, after touching the T cell is true, then this K1 being evaluated to 1. So that means the CRCR4 synthesis will turn on. And then, right, and then that express it, right?
Oh,
so that's how you link that there. Okay.
Okay, if you assign a behavior, if let's see, you have your parameter outlined in rows 400 or whatever in Tellurium, and then your steppable pulls from that to assign a behavior if some condition is met.
Right.
Cool.
Yeah.
Okay.
Right. But in our published version, we're it's more complex because CRCR4, as I said, is being regulated by FOX1. So then the FOX1 is the one driving the expression of CXCR4. Then FOX1 in turn controlled by something else. You can link that, imagine that signal transduction pathways there, right? And then once we need to turn off, here, see in this situation, where in this demo version, we make it quite simplified. We're saying that once the cell moving to the dark zone, as soon as touching the cedar cell, touching the I think this means supposed to be the CRC cells. Okay, once they touching the CRC cells, what have you, okay, it will be able to trigger both mitosis and also, depending on some other variables like kappa B, I'm going to decide to turn off CXCR4 so that you set K1 to 0. Now CXCR4 get downregulated, and then those cells going to be moving towards the lysosome. Okay, yeah. So that's how it's done being here. But then in the published version, it's more complicated. It involves how linked to cell cycle based on some studies showing that CXCR4 will be all expressed as cells as long as the cell in the cell cycle. Once the cell exits the cell cycle, CXCR4 will be downregulated. Okay. Yeah,
you've shown in one slide you simplified the original signaling pathway. Is that what you're referring to, or no?
Exactly. So, for example, the pathway over here. Where is it? I think. Here, right. See this? So for this actual implementation, the CXCR4 can be expressed only when both FOX1 is high, as well as the cell cycle is in motion. Yeah. So
despite high CXCL12 from CRC cells,
it
also has to have It also has to have involvement of FOX1, even despite release of CXCL12. I'm not quite sure how the arrows are acting on the receptor.
Oh, the CXCR12, yes, it's always there, right? It's a gradient, the chemo attractant field in there, right? Yeah. So the CXCR4, oh, actually, I think I know what you're trying to. Okay. So the CXCR4 in our final version of the model is also part of the variables that determine the lambda for the chemotaxis.
Oh, okay.
All right. Yeah. So good questions. Yeah. So in the final version, when you define the lambda stuff here, lambda, that how spell? How do I say lambda? B, right? B. Right. So in our yes, in our final version of that, somehow these lambdas sets is not set to a fixed number. It's set to like proportional to CXCR4 expression as well. For the 12 and for the 13 proportional to CXCR5.
Okay. And then you have your initial species conditions like FOXO1 is set at 100 and kappa B is set at 100 as well. Where are those numbers coming from?
Oh, we try to normalize this between 0 to 100. It itself doesn't mean anything. We can go higher, but when you do Gillespie stochastic simulation, you don't want to go high, too high. 1,000, it can be very costly computationally, and also when you go to 1,000, that doesn't give you enough randomness either. So usually the rule of thumb is when the number of follicles in the system around 100, 200, that's sufficient to generate enough randomness there. Yeah, so that's the way how I do it.
Because there's also, I don't know if you estimated parameters or how you did it, but your K1 through K13, they're just varying from basically 0 or 0.01 or 0.1 or 10.
Okay. Yeah. And that one also determined by the half-life, like the K, for example, the K2 here, right? Determine the half-life of CXCR4. And that K2 is determined by the cell cycle relatively, right? Essentially, in this situation, about 100 MCS, the molecular step is equivalent to 1 hour in biological time. Okay, so once you have that connection, then everything can be determined based on that as a reference, right? Yeah. So apparently, you want to, because these things shuffling like say in one day, they maybe actually make one round or something. Okay, then you have to determine how your CXCR4 half-life going to be. It cannot be too long, right? A few days. Then otherwise, it won't be. If too long, it won't be readily downregulated once it needs to. So usually in that case, we just make CXCR4 like say a few hours or something. Okay, yeah. But what I said about 100 MCS correspond to 1 hour, that's applied to our final version of the model, not to this demo one. All right. Yeah, you don't have to.
Yeah.
Okay. Yeah. So I guess that's about this one here. It will keep just running, and eventually some of these all will be turning red, indicating like high affinity ones there. So now I'm gonna switch quickly to the main script. So I'm running that on I don't know what environment you're using. I'm running that with the Spider. So essentially, if you have a Spider or something similar installed, you can just navigate to the folder where I have this simulation result analysis. And this one has the main script and then have some pre-simulated data sets. Okay. For example, if you open this one, it will actually have everything I said. They have a header, which gives the name of all these variables being saved. And then after that, it's just each line represents, every 15 MCS, how it's being saved. Okay, just like that toward the end of this. Yeah. And every other files has similar structures, exactly same structure. Right. So once you open this, Hayden says that you probably won't be able to run from like a command line easily with this, but he will give instructions later on for you guys to do this offline. All right. So if you cannot run this, don't worry about it. But so essentially, what this script is about reading the file, and then import those into this Python environment. So I'm just quickly running this. Like, say you're gonna import all these libraries or package, what do you call it? And then we have some functions defined to evaluate is a flow number or not. And then we have functions to read files in, all right? And read files saved in the cell data. So here it's kind of like, say you execute this, it's reading everything here, all the TXT files. So that will give you all these files, names. You can see here, right? We have like 267 there. And then, let's just print out how many number of cells. Yeah, you can do this way. And then, okay, if you do this, if you give you the header from those files, so that's produce a header, and that's kind of indicating all these variables that's being saved. Okay, and then here it's I think for this one it's trying to extract all the CDRs, which means has cell which has the generation number 0. So if you run this, if you give it all these, in this case, I have 5 seeding B cells, all right? And then for here, it's another function, as I said, to retrieve that lineage. So that function is being defined. You just need to run this. So that's kind of take this function. And then now you can run, say, do you want to display lineage branches? I say yes. The reason I think we're putting this is because when you have a big data sets, it can take a really while to retrieve all these lineages, branches. So if you run this, it's going to ask you yes, and you say yes, it will be able to retrieve all this. See this now is give you all these lineages, different branches, starting from a CDR cells all the way to some terminal B cells. Yes, each one of those. And then let's say if you want to pick one lineage to display, then you can copy this and then paste over here, say for your chosen branch. So you run this, let's give you the chosen branch. And then this data sets will be able to combine all the data, this chunk of code will combine all the data in each of these cells into one single called branch data here. So that contains all the data sets from that lineage branch all the way to the last cells in there. Okay. And then here it's again, I just for your convenience, it show the header again, because the next thing you're going to be doing is to choose which variables in the headers you're going to be displaying. So, for example, I'm going to like say I want to choose XCOM, YCOM, and then I want to choose the MCS as well, for example, MCS here. So that's my 3 variables to display. And then these are doing some cleaning and the conversion. There's a duplicate headers that have to be removed. So that's what this do. And then now you can do the 2D plotting, which plot the XY. So, here, Is that doing anything yet? Or is, oh, this is the one I have from the previous run. Let me delete this to see. Remove plots, remove plots, 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 remove plots. Right, so if I run this, I'm supposed to be getting my. Oh yeah, right. See, now this one give you that's lineage branch where cell starting from here and then moving back and forth, and the color represents the corresponding MCS in there, right? And then you can also, like say, instead of displaying using this, you can change your Z into something else. So let's see if I can just choose that. Oh, I keep hitting on the wrong one. To CXCR4 here. And then now if I do this again, it will show the, okay, it will show the expression level of CXCR4, as you see over here. Yeah. And then you can do the 3D plot like this as well, X, Y, and CXCR4 levels. Yeah, you can do all the combinations. So this is just really for a very simple demonstration how you do the post processing analysis of the similar data. For the really complicated analysis, as you see in my presentation, for the paper we published, like these ones, population analysis, these ones, those will be available in the GitHub link in our published paper. But I have to apologize that we haven't really clean up and annotate our codes to put on the GitHub there. So it's upcoming, but in the next few weeks, hopefully we'll put everything in there. For those of you who are really interested in the Germinal Center, yes, you can go there to get those scripts.
How were these plots generated, those at matplot libraries? We haven't seen these before yet in the course.
You mean how these are being plotted?
Yeah.
Right, it's not in the one for the demo I just shared with you, but it will be in the GitHub. Okay, that's we promise to put the code for the published paper. Is that what you're asking?
It's an ignorant question I have, which is the plot is drawing from all of this from CompuCell, right? It's just you're using a different plotting code to get these plots.
No, these are not directly from CompuCell3D. It's a post plotting by using those TXT files. If that's what you're asking. These are not directly coming out of the
that's what I asked. Hayden just said the plots are drawn with matplotlib. That's what I asked. I was wondering if those were drawn with matplotlib.
Right.
But you can import matplotlib into here, right?
I think that's what we did. Yeah. You see here, we import these stuff, right? Yeah. So that's what we do. Okay, I think we have until 4:30, right, James?
Yeah, let me put up a poll and then if we'll finish up here. Thank you so much. That was really a fabulous talk.
Yeah, thank you, James, for the invitation. Yeah, again, Hayden says he will provide some additional instructions on how you actually run the Python script over here to to come up with all these figures I'm having here.
Okay, well, I want to thank you. We have time for another few questions if you have any.
Yeah, feel free to ask questions here.
And please if you attended this session, please fill out the poll. and we'll start the next talk at, let's see, what time is it now? It's now 4:30. We'll start at 4:40 for people who want to take a few minutes break, okay?
Mhm.
Can I ask a question, please?
Yeah.
Yeah, hello. I don't know if I understand correctly, but the affinity is calculated from the original sequence at the beginning of the simulation.
Right. Right. You mean how the affinity being calculated, right?
Yeah, but my question is because in some of the step, like this affinity score is used in the SMBL to update the concentration of uh, uh, what's the transcriptor?
Yes. So your question is the SBM model I'm using here?
Uh, because the affinity score affects this transcriptor in I don't know if the FOXO.
I'm not sure I understand your question, Omar.
The way in which the affinity score affects the FOXO transcriptor is one
You mean
over
the
the
FOX1?
sorry,
Are you talking about the FOX1 here?
FOXO1. Yeah.
Uh-huh. So what's the
So
question about FOX1?
the affinity score affects the FOXO1, but it does in 1 over the affinity score or something like that.
Right. I mean, again, this is I just said, okay, this is the intermediate version of our model. So it does not, what you're seeing here does not correspond to the pathway structure I have over here. Okay. Yeah, don't make that connection. It will be our published version of the model. Yes, really reproduce this one. Yeah.
Okay.
Yeah.
Yeah, like the question was the more affinity, the less FOX is produced.
Oh, that's correct. Actually, that's been shown. That's the higher the affinity, the BCR activation will lead to downregulation of FOXO1. That's correct.
So the FOXO1 is like the killer or something like that, like or
No, the FOXO1. So for the MYC activation, I remember it's required both downregulation of FOXO1 and also upregulation of alpha kappa B. Okay, and that will allow MYC to be upregulated. Okay, and that will in turn, induce cell cycle. So, but for the yes, and that also leads to the kind of the so the interesting thing is FOXO1 is only kind of transiently downregulated in the lysosome. So eventually before it's moving to the dark zone, the FOXO1 will get re-expressed. Okay, as a result, the CXCR4 will be regulated, and then the cell will be moving to the dark zone in responding to CXCL12.
Okay.
Yeah.
Okay, thank you.
And then another thing about mix, the mix is also only transiently expressed in the lysosome. However, the B cell will continue to, so the MYC will initiate the cell cycle. However, even though the MYC will downregulate once the cell gets to the dark zone, but it's AP4, which is downstream of MYC, that's responsible for the continuous division in the dark zone.
Whoa. Really? That's like yeah, spatially controlled. That's so funny because it's just missing in the diagram. It just shows high MIC leads to high AP4.
MIC lead to AP4, right? But remember,
Yeah.
the MIC need to be upregulated only once they have in contact with CD40. I mean the TH, TFH, and FDC, right? So essentially, once it's left up the cell left the dark zone, they're no longer being activated by the FDC and the T cell. So the MIC.
Ah,
Right, so the
yeah.
MIC will downregulate. However, AP4, the way I implement, has a very high half-life. So the AP4 will hang around for much longer, okay, which is sufficient enough to allow the cell to divide for like a 2 or 3 round or more. Okay, yeah.
Because that's what's enabling clonal expansion, or no, am I missing that?
Yeah, that's enabling the clonal expansion.
Yeah, it's so funny. My first question was, I was like, wait, if MIC is upregulated and that it leads to proliferation, then why are we not seeing more proliferation in the light zone? But
right. I mean, some people say, yeah, for the B cell in the light zone, they only start to enter the S phase, as far as I understand. Right. So
okay.
yeah, but then once it migrates to the dark zone, they'll start to divide. Yeah, but there's some conflicting literature. Some people say, okay, there's some kind of a division happening in light zone as well. But I think the majority of the literature, the consensus tend to say is 90% of it happening in a dark zone. Yeah.
So interesting. Thank you so much.
Yeah, you're welcome.
I want to thank you again. I'm afraid we're going to have to stop now and move on to our final talk of the day. I'm really delighted that we had such a stimulating discussion on this one. And I'm glad that many people were able to get it to run. It's exciting to see such a detailed model that has so much important biology in it. So I want to thank you again. I'm going to.
Okay. Thanks, James. Yeah.
Thank you. I'm going to take just a second to set up for the next talk.
Sure.
And
yeah.
so thank you so much.
You're welcome.